How about sauna and board games tomorrow?
In the evening? That would work, yeah.
Yeah.
And that will probably not work for me.
Oh.
I have a scheduled panic attack for tomorrow. I have to go for some B12 injections. So.
That's a bummer.
Don't you have vitamin supplements by the injections? Or could you just eat meat?
Well, technically I could if my body was able to absorb the vitamin B12. But there's this protein called intrinsic factor which binds the B12, and your stomach kind of recognizes the intrinsic factor and then it's able to take the whole B12 in. But I have pernicious anemia, so my body actually attacks the intrinsic factor. So I don't get any absorption even if I dose a very high amount of vitamin B12.
Yeah.
Okay.
Do you know how vitamin B12 absorbs?
No.
Okay, let's go through it then. Have you ever wondered how vitamin B12 gets absorbed? Meat. Meat contains vitamin B12. Here is your stomach. Your stomach contains stomach acid and a protein called haptocorrin. Meat enters the stomach, and stomach acid releases B12 from the meat. B12 can now bind to haptocorrin, which protects us from the stomach acid. Stomach cells also produce a protein called intrinsic factor. The B12 haptocorrin complex and the intrinsic factor move into the duodenum, where pancreatic trypsin, an enzyme, breaks up the B12 haptocorrin complex. B12 now binds to the intrinsic factor. Together they move through the intestine until they come to the ileum. In the ileum, the B12 intrinsic factor complex binds to the Cuban receptor. The B12 intrinsic factor Cuban receptor complex is taken into the cell, where cellular enzymes release B12 from the receptor. And now we know how B12 absorbs.
Oh wow, I didn't know that.
Yeah, for people with absorption issues or pernicious anemia and stuff like this. For us, actually, the only current treatment that works is lifelong B12 injections, which really sucks because it hurts, and a lot of us have like fear of needles, and there's all the hassle of like having to book doctor's appointments and like the whole accessibility thing, and then also there's like the whole other aspect of like environmental waste.
That's why our Aalto-Helsinki iGEM team is working on this
Really?
year. Yes.
What are you doing?
So what we are trying to do is to create new generation oral B12 supplements to help people who has absorption issues like you.
Oh yeah?
Think about it. AbsorBuddy, new generation oral B12 supplement, which contains both B12 and intrinsic factor. So in our lab, we are trying to produce intrinsic factor proteins in yeast by using 5 different species, and after that comparing their efficiency from different angles, like how well they bind to B12, what are their antibody interference, and what are their structure. Only with this we can find the best candidate to be used in next generation supplements.
Oh, that sounds so fancy.
Yes, but to make it meaningful, we first needed to understand how is this important for people and how this type of new oral supplement would fit into the real world.
Yeah.
And that's where our human practices come in.
Vitamin B12 is essential for red blood cell formation, neurological function, and DNA synthesis. Yet, millions of people worldwide struggle to absorb it properly, whether due to autoimmune diseases like pernicious anemia, gut disorders, surgeries, certain medications, or simply aging. Diagnosing vitamin B12 deficiency is far from simple. Its symptoms, fatigue, memory loss, confusion, difficulty walking, can easily be mistaken for other conditions, and standard blood tests don't always detect it. On top of that, the threshold for what counts as deficient varies from country to country, meaning that some people may still suffer from deficiency even when their test results appear normal. Because of these inconsistencies, we don't actually know how many people are affected globally. Estimates range widely from 2.6% to as high as 26% of the global population could have B12 deficiency, and between 0.1% and 0.8% could have pernicious anemia. What struck us most, however, was how little recent research exists on B12 malabsorption and possible treatments. In particular, studies on intrinsic factor, the protein essential for B12 absorption, largely date back to the 1950s, leaving major gaps in understanding.
Yeah, that must have been confusing.
Yes, so to bridge this gap, we decided to go beyond the lab and listen directly to those living with the condition.
And who did you talk to?
We began by speaking with patients and advocacy groups, including the Pernicious Anemia Society. These conversations revealed that while intramuscular B12 injections remain the most effective treatment, they come with major challenges. Each dose requires emotional and logistical preparation. It's painful, time-consuming, and requires patients to plan their lives around clinic visits. The process is especially difficult for those with limited mobility or living far away from healthcare centers. Even in countries with public healthcare, like Finland, the hidden emotional and financial costs quickly add up. Beyond these issues, we learned that rigid treatment guidelines can ignore individual needs. The standard 1 injection every 3 months doesn't work for everyone. Yet, asking for more frequent care can lead to disbelief and even conflict with doctors, forcing some patients to fight for recognition.
Really?
Many described years of misdiagnosis or dismissal before receiving proper treatment. This often results in continuous severe symptoms and sometimes permanent nerve damage.
What did you do then?
We also interviewed researchers and medical professionals who echoed many of these concerns, confirming both the scientific and clinical gaps. They also noted how injection-based treatments put a strain on healthcare systems and generate significant environmental waste due to syringes, needles, and plastic packaging.
What was this needed for?
These insights shaped our project vision towards designing something that gives patients autonomy, reduces suffering, and hidden costs. With an oral supplement adapted to certain types of B12 malabsorption, more patients could manage their treatment independently without constantly having to justify their needs to doctors or plan regular travels. This new solution would also be more accessible, reduce strain on healthcare systems and their environment.
Our lab methodology began with the design and construction of expression vectors for intrinsic factor proteins from 5 species: human, bovine, rat, platypus, and porcine. Each gene was codon optimized and cloned into a yeast-compatible vector with an alpha factor secretion signal and histag-enabling protein secretion and purification. We confirmed successful assembly by colony PCR and sequencing. After transforming Comagatella puffy yeast, we screened multiple colonies for intrinsic factor expression. STS page analysis revealed strong expression for bovine and rat intrinsic factor, while human and platypus intrinsic factor showed lower yields. For purification, we adapted from automated affinity chromatography to a manual gravity flow method due to equipment failure. That incident slowed things down for 1 week. Size exclusion chromatography further refined our samples, and we selected the purest fractions for our downstream assays. Porcine intrinsic factor was later excluded from analysis stage due to insufficient recovery after purification. To assess function, we developed a UV-vis binding assay to quantify B12 binding efficiency. Bovine intrinsic factor demonstrated the highest binding, while rat intrinsic factor performed poorly, and platypus intrinsic factor showed moderate binding.
Ooh, hey, that sounds exciting.
Yeah. We also performed indirect ELISA to evaluate antibody cross-reactivity. Human intrinsic factor showed strong binding to anti-human intrinsic factor antibodies, while bovine and platypus intrinsic factors had reduced recognition, suggesting potential to evade immune interference.
Wow, that was significant.
For structural validation, we used circular dichroism spectroscopy, confirming that bovine intrinsic factor adopts a correctly folded, mixed alpha helical and beta sheet structure. In the dry lab, we compared binding affinities of these intrinsic factors with B12. Non-human intrinsic factors were predicted with AlphaFold, relaxed by MD in water, and submitted to docking test. We aligned all proteins to human intrinsic factor B12 complex and ran glide SP docking within a conserved 6 angstrom pocket. Platypus gave the most negative docking score, but bovine showed the tightest, most stable packing. Porcine was the weakest. Overall, bovine and human form the most structurally robust intrinsic factor B12 complexes. And it was really exciting that dry lab and wet lab results were consistent with each other. In conclusion, our multi-assay workflow revealed bovine intrinsic factor as a strong candidate for oral B12 supplementation, combining structural stability and robust B12 binding.
Everything fit into pieces. Our project brought together synthetic biology and patient insight to create the foundation for a novel oral vitamin B12 supplement, one that could drastically improve the lives of those affected by B12 malabsorption and pernicious anemia. In the lab, we successfully produced and compared intrinsic factor proteins from 5 different species. Among them, bovine intrinsic factor emerged as the most promising candidate, showing both strong B12 binding and signs of lower immune reactivity. This combination opens a potential path towards creating an oral supplement that is not only effective, but also patient-friendly. What makes this achievement meaningful goes beyond the experimental results. By integrating feedback from clinicians, researchers, and patient communities throughout our process, we ensured that our scientific direction stayed aligned with real human needs: accessibility, autonomy, and quality of life. Looking ahead, our work lays a foundation for future development. Further structural and receptor binding studies could deepen our understanding of how intrinsic factors from different species interact with vitamin B12. Optimizing protein expression and expanding experimental validation will be key steps for future teams and researchers seeking to build on our findings, moving the field closer to practical applications and bridging the research gaps surrounding intrinsic factor and B12 absorption. Equally important is the collaboration beyond the lab. Our work was made possible through the contribution of universities, institutions, companies, and mentors whose expertise and support shaped every step of our project. We promoted accessible science communication through workshops and informative videos that brought synthetic biology closer to the public. These activities highlighted the importance of education and accessibility in science. Looking ahead, we continue sharing our work through upcoming events with our collaborators and sponsors, including a pitch talk at Y-Science, a side event of Slush, strengthening the bridge between research and innovation. Our project demonstrates how student-driven research can make significant progress toward medical innovation, showing that with creativity, interdisciplinary teamwork, and a commitment to patient-centered approach, even a small team can make a meaningful impact on future healthcare solutions.
Wow, that sounds like a lot of work.
Yeah.
I always thought that student projects are really small, but this could potentially improve people's lives.
Yeah, absolutely. Especially for people with pernicious anemia or B12 absorption issues, which are, by the way, incredibly under-researched. Any new knowledge that is brought towards possible solutions is always towards home. So this really means a lot that you're trying to take on this project and this whole operation.
Yeah, and for us it was never just a scientific challenge. It really means and matters to us that this helps your life in a better way, and also that we can open up these conversations about this.
Yeah, we don't have all the answers yet, but we believe that this is the first step towards building a healthier future.